Hello, welcome to another screencast where this time I will be discussing, if I can get this to work, lipids and some characteristics there of lipids. And so hopefully by the end of this you should be able to identify and describe here the structure of specifically triglycerides, right? So what a triglyceride is, and then some other examples of lipids. We're going to then look at the structure of cholesterol and be able to identify both of these things as lipids, but being able to see how they are actually completely different from one another. And then looking at enzymes here associated with lipid metabolism, being able then to explain here some of the metabolism of lipids, and again the importance of lipids to cells, right? And again why do we even care about any of this. So let's get started here. And now we are on the far right side of this diagram here, where the fats, which should also be colored yellow here, so fats also sometimes you'll hear me refer to them more often here as lipids. Primarily the form of the fat that we ingest the most are going to be these triglycerides, right? And the triglycerides are broken down into fatty acid components as well as then these glycerol components here. Unlike the carbohydrates and the proteins over here that we covered in other screencasts, which have these polymer, you have this polymer here, then broken down into individual monomers that then get absorbed directly into the bloodstream. There's a little bit of a different vibe going on over here with the lipids. They get broken down in these fatty acids and these glycerol, which then get absorbed into the lymphatic system at a lacteal in the villi. And they are then, to various degrees, reassembled and then packaged here, and then loaded into the bloodstream and transported thusly. So there's a little bit of a different theme going on over here, which we can talk about in other screencasts as well. But for now, let's talk about here the triglycerides. Again, the most common form of lipid that we ingest, and they are made up of these glycerol molecules and these fatty acids. A glycerol is this molecule here in gray, which is a C3, so there's the 3 carbons. And then you have hydrogens here, 1, 2, 3, 4, 5, 6, 7, 8, not shown there, and then O3. So this is a glycerol molecule, and it is going to be chemically bonded right here to 3 fatty acid tails. Now these fatty acid tails, you notice that what I want to point out here is that they are primarily hydrocarbons. So a hydrocarbon tail here that's made up of simply carbons and hydrogens. Yes, I know at this end they have this oxygen on this carboxyl group over here, but this is not really the reactive part of the molecule over here. It's these fatty acid tails that make this molecule extremely nonpolar, right? That is, it will not react with water, will not be dissolved in water or by water, right? Which makes it difficult if you're ingesting them, because into the stomach, most of the secretions there in the stomach are in aqueous environment. The secretions in the small intestine are in aqueous environment, so in water, aqueous environment there. And so you would have these nonpolar fats, nonpolar lipids not being dissolved, which makes it difficult for metabolism, but we will talk about how that is dealt with a little bit later. So a little bit more on these fatty acids here. The fatty acids, you've probably heard of saturated fat and then unsaturated fat. So here's an unsaturated fat, here's an unsaturated fat over here. And it all has to do with the location of the hydrogen. So in a saturated fat, you have between the carbons of the hydrocarbon there, you have only single bonds, but in an unsaturated fat you have a double bond between the carbons there, right? So saturated fats here tend to be solid at room temperature. These are things like lard or animal fats, right? So solid at room temp, and this is animal fat. Right? So if you fry up bacon in a pan and leave that, or you're frying up ground beef in a pan and it's, you know, the ground beef is 85% protein and 15% fat. The fat that is left in the pan is going to be saturated fat. Right? And if you let it cool down, it's going to turn all white and solid, and that is what saturated fat is. And again, we're looking here at the fatty acid tails. An unsaturated fat, such as this one right here, is liquid at room temperature, and these are your oils, right? For example, olive oil, corn oil, peanut oil, right? Etc. So any oils tend to have this unsaturated fat right here and have these double bonds. Now there's a little bit of an issue here with this is technically an unsaturated fat, but this is the trans fat. So you've probably heard of trans fats. And so a trans fat here looks like which one? Looks like the unsaturated, looks like the saturated. Well, it's pretty straight here, so it looks like this saturated one over here. So trans fats actually tend to chemically behave very similar to these saturated fats. And so this is why we've sort of removed trans fats from foods in favor of these unsaturated fats here. And that was just kind of a fun fact, but there's the difference between the saturated and the unsaturated fats. Now, earlier I talked about these lipids being nonpolar and being difficult they will not dissolve in the aqueous environments of the stomach or of the small intestine. And so what would happen is any fat that you took in would form this really large fat globule here. Now this has a very small surface area to volume ratio, and so if you had enzymes that were going to be digesting this fat, and the enzymes that do that are lipases. These enzymes would be digesting, chemically breaking down the glycerol and the fatty acids from the edge right here, and it would take a long time. So what we've done, or what happens is the bile salts that come out of the gallbladder here, and these represent the bile salts here. The bile salts help to emulsify the fat. And so these bile salts will break this large fat globule up into smaller fat globules, and the mechanical digestion of the stomach and the small intestine helps to churn this up and keep the fat globules separate, much as what we're seeing up here. So if you had oil and vinegar in a flask and you let them go, they would not mix. And if you then shook it up here, you get these individual little particles of the lipids, and if you let it sit back down again, those lipids would tend to reform with one another, and then you get that separation again. Well, what happens with these bile salts is that the bile salts, the mechanical breakdown, mechanical digestion in the stomach sort of churns it up here, and churns up into all these small pieces, and then the bile salts get in there and will surround all of those fat globules, and then the bile salts prevent, so this emulsification is the prevention of large globules reforming. And so this this emulsification helps to increase the surface area to volume ratio of these fat globules, and that aids in lipases being able to break down these triglycerides into glycerol and fatty acid much more quickly and efficiently, and also allows for the transport of these things into lacteals, etc. So switching gears completely there, again, that was all triglyceride information. There's another class of lipids called the steroids, right? And so steroids up here are unique lipids based off the structure of cholesterol. So cholesterol is a steroid, and this cholesterol molecule is also extremely nonpolar. It has this hexagon, pentagon backbone structure there with these different chemical groups sticking off of it. And again, this looks nothing like a triglyceride. This is something completely separate, but gets thrown into the lipid category because they are nonpolar in nature. Right? And so cholesterol is important in the diet, and we talk in another screencast about the processing of cholesterol, and you often hear HDL and LDL here in terms of cholesterol transport and everything. But some cholesterol is important because you have things like vitamin D, which are derived from this cholesterol, and you can see here the remnants of this hexagon sort of right there. Sorry, of this hexagon right here. And so vitamin D is derived from that. You also have other steroid hormones here, cortisol, right, aldosterone, and then your testosterone, estradiol, estrogens, right, progesterones. These reproductive hormones are also all derived from cholesterol here, all this basic structure. So some cholesterol is important for these things, and also not shown on this diagram, cholesterol is important for cell membranes. It is a key role, it plays a key structural component of cell membranes, and all cells have cell membranes, and not only cell membranes but also the membranes of mitochondria and things like that as well. So cholesterol, pretty important molecule, but in large concentrations can be an issue, and again we'll look at another screencast where we talk about the processing of cholesterol there. So let's go back to the triglycerides here again. The unemulsified fats, the triglycerides, are what we take in, and so the emulsified by the detergent action of the bile salts, and again this is what detergents are doing. Detergents are breaking up the surface tension, making it easier for them to be dissolved. And so ease of dissolving in water. And this is where the enzymes are. So the bile salts from the liver, this emulsification takes place, and then you have this pancreatic lipase, which is going to break down the triglycerides into fatty acids, here the individual fatty acids, as well as then into glycerol. Right? And then the absorption of fatty acids and monoglycerides enter, again, we said the lacteals here, that lymphatic system, right? And then are eventually going to be transported into the bloodstream and into the liver as well. So again, here we go with this is what we're talking about. Here's the triglycerides. So now you should be able to identify this part right here, the glycerol. Each of these would be the glycerol part, and then here are the fatty acids there. And again, here's our fatty acids and our glycerol molecule that is going to be reassembled to some point here, to some extent in the lymphatic system, and then transferred ultimately into the bloodstream from the lymphatic system. Now, why are fats important? We talked about cholesterol is important, but why are the triglycerides important? Well, the main cellular use of triglycerides here is that fats are the primary fuels for many cells. So there's an energy molecule component here to them once they get inside the cells. Another is a structural role. Fats build myelin sheaths and the cell membrane. So the myelin sheaths around, again, the neurons. Right? So fat is important for these reasons. And also it plays a protection role and an insulation role, fat does. Right? And so this is why these triglycerides that we ingest over here, again the glycerol and the fatty acids, transported to the liver, metabolized by the liver, and we'll talk about this in another screencast over here, but then this is why they're important in cells. Okay. So again, you should be able to identify here, describe here the structure of triglycerides as well as cholesterol, sort of compare those two. Some examples of lipids in terms of animal fats and oils, and then the enzymes, and so not only the lipase, but then also here the bile salt, which is not an enzyme, but helps with the emulsification of the fat. And why is that important in the stomach, or sorry, rather in the small intestine? Right? And then a little bit about the metabolism, the breakdown there, and of course then the importance of lipids to the cells, and that again is the triglycerides as well as the cholesterols. Okay, so hope that was helpful. Thanks for listening. Bring your questions to class.